Hello, and welcome to your Anatomy Physiology Bio 202 video lecture on blood. I'm Mr. Kennedy, and I'll be your guide as we explore this topic together. Blood is a liquid connective tissue consisting of cells surrounded by a liquid matrix known as plasma. The plasma makes up 55% of the total blood volume. The suspended solids, or red blood cells, make up the remaining 45%. The cellular components, or formed elements of blood, include red blood cells, white blood cells, and platelets. The plasma portion of the blood consists of water, proteins, and other solutes. This table illustrates the components of your blood and their relative proportions. And this is what your blood might look like under a microscope or a scanning electron micrograph. Blood transports oxygen, carbon dioxide, nutrients, hormones, heat, and waste products. Blood regulates homeostasis of all body fluids, pH, body temperature, and water content of cells. Your blood protects against excessive blood loss by clotting and against infections through the use of white blood cells. This table illustrates the substances in the blood plasma, giving a brief description and a function of each. Lymphocytes. Lymphocytes are able to live for years, while most other blood cells live for hours, days, or weeks. The number of red blood cells and platelets remains rather steady, while that of the white blood cells varies depending on invading pathogens and other foreign antigens. The process of producing blood cells is hemopoiesis, or hematopoiesis. Pluripotent stem cells differentiate into each of the different types of blood cells. This chart illustrates the potential for a pluripotent stem cell to differentiate into a wide variety of formed elements. The red blood cell, or erythrocyte, contains the protein hemoglobin that is used to carry oxygen to all cells and to carry 23% of total carbon dioxide to the lungs. Each hemoglobin molecule contains an iron ion, which allows each molecule to bind 4 oxygen molecules. Red blood cells have no nucleus or other organelles and are biconcave discs. The lack of a nucleus and the shape allow the cells to efficiently carry oxygen. This is an illustration of a red blood cell and the hemoglobin molecule. Hemoglobin is also involved in regulating blood flow and blood pressure via the release of nitric oxide, which causes vasodilation. Vasodilation improves blood flow and enhances oxygen delivery. Red blood cells also contain carbonic anhydrase, which catalyzes the conversion of carbon dioxide and water to carbonic acid. This compound transports about 70% of the carbon dioxide in the plasma and can also act as a buffer. Red blood cells live for only about 120 days. Dead cells are removed from circulation by the spleen and liver. Breakdown products from the cells are recycled and reused. This is an illustration of the life cycle of a red blood cell. Erythropoiesis, or the production of red blood cells, begins in the red bone marrow. Reticulocytes, or immature red blood cells, enter the circulation and mature in 1 to 2 days. Erythropoietin, a hormone released by the kidneys in response to hypoxia, stimulates the differentiation of hematopoietic stem cells into erythrocytes. This is an illustration of the feedback mechanism that controls the production of red blood cells. White blood cells, also known as leukocytes, contain a nucleus and organelles, but no hemoglobin. Leukocytes are classified as either granular, containing vesicles that appear when the cells are stained, or agranular, containing no granules. Granular leukocytes include neutrophils, eosinophils, and basophils, while agranular leukocytes include the lymphocytes and monocytes. This is an illustration of what each cell type might appear like under a microscope. White blood cells may live for several months or years. Their main function is to combat invading microbes. During an invasion, many white blood cells are able to leave the bloodstream and collect at sites of invasion. The process is called emigration or diapedesis. This is an illustration of diapedesis. In general, an elevation in white blood cell count usually indicates an infection or inflammation. A low white blood cell count may develop due to several causes. A differential white blood cell count will help to determine if a problem exists. This table summarizes the significance of high and low white blood cell counts for each cell type. Platelets. Platelets are used to clot the blood. Under the influence of the hormone thrombopoietin, hematopoietic stem cells differentiate into platelets. Megakaryocytes in red bone marrow splinter into 2,000 to 3,000 fragments to create the platelets that contain many vesicles but no nucleus. Platelets survive for only 5 to 9 days. This table provides a summary of the formed elements in blood by type, including their number, characteristics, and function. Stem cell transplants from bone marrow and cord blood. Bone marrow transplants are performed to replace cancerous red bone marrow with normal red bone marrow. The donor's marrow is usually collected from the iliac crest of the hip bone. Stem cells collected from an umbilical cord after birth are frozen and may also be used and have advantages over bone marrow transplants. Hemostasis. Hemostasis means to stop bleeding. The process involves vascular spasm, platelet plug formation, and blood clot, or coagulation. This is an illustration of hemostasis. Blood clotting involves several clotting or coagulation factors identified by the Roman numerals and divided into 3 stages. The 3 stages are the extrinsic, intrinsic, and common pathway. This diagram illustrates the 3 stages of hemostasis. And this table denotes the clotting factors that are used in hemostasis by number, by name, by source, and pathway of activation. Once the clot forms, it consolidates or tightens to pull the edges of the damaged vessel together. Vitamin K is needed for normal clot formation, although it is not directly involved. It is used in the synthesis of 4 clotting factors. Small, unwanted clots are usually dissolved by plasmin. Blood groups and blood types. Blood is characterized into different blood groups based on the presence or absence of glycoprotein and glycolipid antigens, also known as agglutinogens, on the surface of red blood cells. There are 24 blood groups and more than 100 antigens. Because these antigens are genetically controlled, blood types can vary amongst different populations. Classification is based on antigens labeled A, B, or AB, with O being the absence of the antigen. An additional antigen, Rh, is present in 85% of humans. This table designates blood types in the United States by population group and frequency. Blood plasma usually contains antibodies, or agglutinins, that react with A or B antigens. An individual will not have agglutinins against his or her own blood type. This is an illustration of the blood types with their antigens and antibodies associated with each. And this table will provide you with a summary of the ABO blood group interactions. In order to determine a person's blood type, typing and cross-matching are performed. A drop of blood is mixed with an antiserum that will agglutinate blood cells that possess agglutinogens that react with it. This is an illustration of how blood is typed. Hemolytic disease of a newborn. At birth, small amounts of fetal blood leak into the maternal circulation. If the baby is Rh positive and the mother is Rh negative, she will develop antibodies to the Rh factor. During her next pregnancy with an Rh positive baby, when she transfers antibodies to the fetus, which is a normal occurrence, she'll be transferring anti-Rh antibodies that will attack some of the fetus's red blood cells, causing agglutination and hemolysis. Disorders and homeostatic imbalances. The final thing to note about blood is that there are some homeostatic imbalances experienced associated with it. Sickle cell disease is a genetic anemia. In this case, red blood cells of individuals with this disease contain hemoglobin S that causes the red blood cells to bend into a sickle shape, which gives up oxygen to the interstitial fluid. This is an illustration of a normal versus a sickled red blood cell. And this concludes our coverage of blood.